Okay, so where we left off in the last video was just the overview of acquired immunity and how it's broken into two categories: humoral immune response and cell-mediated immune response. If you recall, the humoral was the activation of the B cells and how they divide and differentiate to give rise to plasma cells, which will secrete antibodies, and memory B cells. And so the reason why they call it humoral, it's right here, because it involves substances found in what's called the humors. They used to call the body fluids humors. So I'm going to put in parentheses body fluids. So the antibodies are secreted into the fluids. They can go to the bloodstream, and so on and so forth. And so it depends, and the extracellular fluid, this fluid surrounding your cells, and therefore can attack and bind to the pathogens that are invading your body. So that's why it says it happens in the extracellular fluid. So it secretes antibodies that defend against pathogens and toxins in extracellular fluid, and your B cells will be there to elicit a very fast response the second time you come into contact with that pathogen. And so what we're going to look at is we're going to move into cell-mediated response, but we're going to look at this helper T cells now. When it says first exposure to antigen, notice that the arrow actually doesn't go right away to the B cells. It says the antigens are engulfed and displayed by dendritic cells. These guys, dendritic cells, it's right here, these are a type of phagocytic cells. So they phagocytic cell. So they undergo phagocytosis and present the antigen. And so antigen is engulfed and displayed by dendritic cells. So notice here, back over here with the helper T cell, when we looked over at this, the antigen presenting cell, which is part of the immune system, so that could be a macrophage, it can be a dendritic cell, is an antigen presenting cell. It engulfs the microbe and, remember, displays part of the antigen on the surface of it with these MHC molecules. And then a helper T cell. Now there's different kinds of helper T cells. The matching helper T cell that has the receptors on the surface to bind with that particular antigen will bind to that. And that's what activates your helper T cells. So we're going to see here that the helper T cells actually are what activate the B cells to actually divide and differentiate into plasma and memory cells. So we're going to build on what we talked about here and talk a little bit about the helper T cells. So they activate by releasing molecules called cytokines that activate B cells, and then they divide and differentiate to become more active, to divide and have more active helper T cells to activate more B cells, and also have memory cells. Okay, so let's look at this. So helper T cells, a response to nearly all antigens. So that's what this is here. It activates the helper T cells. A surface protein called CD4 binds the class II MHC molecule. This binding helps the helper T cell join to the antigen presenting cell while activation occurs. And I'll explain that in this picture here. Activated helper T cells secrete what we call cytokines. These are chemicals that stimulate other lymphocytes. So if we look here, this figure right here, this is the figure in your book. This is an extension of figure 43.9, part B. That is that picture that I just talked about over here. So this picture right here, there's 43.9 right there, and this is B. And so you have a microbe engulfed by an antigen-presenting cell. It presents the antigen, and the helper T cell is binding to it. So why I said this is an extension is it's showing you the exact same type of thing. If we just cover up this part of the diagram and just look at this part here, this is your dendritic cell. And your dendritic cell, let's put this in a color here. So here's your dendritic cell. This is This is a phagocytic cell. This one's phagocytic cell, and it is also an example of an APC, the antigen presenting cell. And so it's going to take in the bacteria, right, by phagocytosis. That's why it's a phagocytic cell, and it displays the antigen. So here's the antigen being displayed right here, and so that's why it's an antigen presenting cell. Then notice here, here's your helper T cell binding to it, and it helps to bind this. See this little guy here, this little arm, the CD4 binds to the MHC molecule, so it binds there. So they're binding together. So that part of the diagram is just like this part. Just giving you a little bit more detail. So the two are binding, and so that's that part. So then you see here that the dendritic cell, the antigen presenting cell, is releasing a chemical. That's this little guy here, releasing a chemical that's going to communicate with the helper T cell. That chemical that it's releasing is called a cytokine. So that cytokine is going to activate the helper T cell. And if you look here, what happens when it stimulates the helper T cells produce and secrete their own cytokines. So that's what we see here is this arrow here, and the cytokines, they release cytokines here. And these cytokines activate your B cells and your cytotoxic T cells. And so look at what your B cells, when it activates the B cells, it activates the B cell to divide and differentiate and eventually make memory cells and plasma cells. And the plasma cells will secrete the antibodies. And the cytotoxic T cells, these are involved in cell-mediated immunity. We're going to look at here that they're involved in this part here, that helper T cells activate B cells, but then they also activate over here the cytotoxic T cells. Let's focus here on the B cells for a second because we're on that trend with B cells. So let's write the different steps here. There are 3 different things here. So step 1 here, let's just write about that down here. So step 1, what's going on there? The helper T cell and the antigen presenting cell, or APC, join. And then what does the APC produce and release? It releases cytokines. APC releases cytokines. So that's step 1, and we can underline maybe cytokines here in pink, since we highlighted them in pink here. So that's what's going on in step 1. So then let's highlight step 2. Let's do purple here for step 2. So here's my purple. Step 2. What's going on here? Step 2, the cytokines are released, the binding to the APC and the cytokines being released by the APC stimulate the helper T cells to make their own cytokines. And so let's write that here. So binding to the APC and the cytokines released by the APC. And again, you can write that out if you don't know, you're not going to remember what APC stands for. Stimulate your helper T cells. Which release cytokines. Okay, and we can underline all the cytokines again in pink. Now step 3, let's talk about step 3. Let's do red for step 3 here. So I'm going to write something that's not shown. When the helper T cells become activated, they not only produce cytokines, but they divide and clone themselves. So I'm going to put here two things for this. Active helper T cells clone themselves. I'll put in parentheses that this is not shown. So when these guys become activated by binding to the ATPC and the cytokines chemically signaling them to trigger it to do something different. So this is the chemical signaling process where it binds to the receptors, signal transduction pathway, and it triggers it to release and make and release its own cytokines, as well as divide to clone themselves. And then the second thing here is the cytokines released by the helper T cells. Cytokines released by helper T cells stimulate two types of cells, right? Stimulate cytotoxic T cells and B cells. So that's what's going on here. So that's what we just looked at down here is this helper T cell becomes activated and it's going to stimulate B cells to divide and differentiate, and it's also going to stimulate cytotoxic T cells. And then this arrow to itself is it's going to communicate with itself and cause itself to divide and differentiate and make more helper T cells that are active, as well as some memory helper T cells. And so that's what those three arrows are here. So let's look at this part here with the B cell. So, so let's write here. Let's get me a pen here. So these guys here with the B cells, when it says they activate the B cells, the cytokines, and it says you're going to eventually get secretion of antibodies by the plasma cells. Let's go to figure 4317. Whoops. 4317. Let's skip over to that. So it's not the next page, but it's the following page, the top of the next page. So let's look at this because it is showing you exactly what I just talked about here. So this is the antigen presenting cell. Here's your helper T cell that binds to it. The helper T cell binds to it, and now you have all these activated helper T cells, so it divides and differentiates. And these activated helper T cells make and release cytokines. And the cytokines stimulate the B cells. And so this part right here is when it activates the helper T cell is this part right here. The antigens engulfed and displayed by the dendritic cells activate the helper T cell. Here are your antigen displayed by the dendritic, or in this case it's a macrophage. It's some kind of phagocytic cell that will present the antigen. So this is your antigen presenting cell. So we can say for this, say here, this is the antigen presenting cell. Presenting the antigen activates the helper T cells. Helper T cells release the cytokines, and the B cells are now activated. And what do? They divide and differentiate. Clones. They clone the plasma cells, which secrete antibodies, and they clone the memory cells, which remember are the long-lived parts that are going to be helpful for the second infection. And so that's what this here is. And so these B cells that become activated, when we said that you get a pathogen, you have to go through that clonal selection process to select which B cells. Helper T cells help to select the B cells. So it activates a particular type of helper T cell that binds to a certain type of B cell and activates only that B cell a particular shape on it. So that kind of goes with that. So that's your B cells there, and how that the helper T cells help with that. So we're going to come back to this in a little bit here as we go along, but just wanted to explain how that happens. All right, and we can write, if we go through right before this picture, we can write a couple of things about that, because it says B cells are response to extracellular pathogens. So the activation of B cells is aided by blank and an antigen binding to blank cells. And so that is, we write here, aided by cytokines and the antigen binding to helper T cells. Whoops, T. So the clonal selection process of B cells generates antibody-secreting plasma cells, and the effector cells of humoral immunity. So the antibody-secreting plasma cells, your plasma cells, are the big things that's going to play a role in fighting the current infection. Okay, so let's go back to this here. So we've talked about the B cells being activated with the help of the helper T cells. The helper T cells activate themselves, which by releasing cytokines, and triggers them to divide and differentiate for active and memory helper T cells for the memory for your secondary infection. So now those helper T cells can secrete cytokines to activate your cytotoxic T cells. So let's just talk about this part. This is called cell mediated immune response. This is a little bit different. The outcome, the purpose of the T cell, the cytotoxic T cells, is to kill the infected cell. Notice here at the end, it says defend against infectious cells, cancer cells, and transplanted tissues. So basically, it will kill off cells, human cells, versus the humoral immunity with the B cells was to secrete antibodies that defend against the pathogens. And so in this case here, it's not against the pathogens, it's against the infected cells here. So that's a little bit different here. So I can add to this when it says, when the cytotoxic T cells are activated here, it says cell-mediated immunity, and it says attack infected cells. So this guy will goes and finds an infected cell and then basically kills it. So how does that happen? So let's look at the cytotoxic T cells, a response to infected cells as well as cancer cells. So cytotoxic T cells makes a protein called CD8, it's on the surface, that enhances interaction between a target cell and a cytotoxic T cell. Binding to a class 1 MHC complex on an infected cell activates a cytotoxic T cell and makes it an active killer. So the fact that these MHC molecules are on the infected cell activates that cell. All right, and so this, let's put here figure, what figure is that? Figure 43-9A. So if we go back to that, 43-9, we just looked at B. Here it is. Here is an infected human cell. Remember, it also displays the antigen fragment, and that's how it knows that it is infected. So this is the abnormal human cell. So then cytotoxic T cells, which are very specific to specific antigens, bind to that, and that's what activates the cytotoxic T cell. So that's what we just wrote here, right here, that it activates a cytotoxic T cell by binding to the class 1 MHC complex. So therefore, going back to that picture, the MHC complex doesn't go on the surface until the cell is infected, and so therefore, that's how it knows it's an infected cell. So the activated cytotoxic T cell secretes proteins that destroy the infected target cell. All right, so let's look at this here. So here you have your target cell. This is your human cell. And so we can say that this is your infected. So this is an infected human cell, let's say, since we're talking about the human, but it doesn't have to be, but we're talking about the human immune system. So here's your target cell. The target cell presents the antigen. So here's your MHC molecule presenting the antigen. And notice here, then your cytotoxic T cell. So this has already been activated. The cytotoxic T cells, let's write down below why they're activated. So notice here, let's do again step 1 here. So step 1 here, the cytotoxic T cells activated by, do you remember what they're activated by? 2 things. Cytokines from the helper T cells, so that is, going back to this picture here, helper T cells secrete cytokines and activate the cytotoxic T cell. And in addition to that, the binding to the target cells. And 2, the second thing, binding to target cells. And so then they become active. Remember we said that binding to the MHC complex on an infected cell activates a cytotoxic T cell and makes it an active killer cell. So they then become active killer cells. And so that's what we see here. All right, in this step. So then here, let's look at step 2. So step 2, what's going on? So this is now activated. So what it does is produces that molecule. It produces a molecule called perforin. So perforin, right here, is released. And what it does is triggers pores to be put into the membrane of the infected cell. All right, so let's write that. So the active cytotoxic T cells release 2 things. They release perforin, which in parentheses, let's write that it forms pores in the target cell's membrane. And the second thing is it releases enzymes to trigger cell suicide, apoptosis. So this is actually right here, step 3. So kind of both of those are red there. So if we look here, the perforin pokes holes in them, and the enzymes trigger the cell to undergo apoptosis. Notice here the cytotoxic T cell is released, and it can be reused, and the infected cell dies. So therefore, that's what's happening here. So when the cytotoxic T cell is activated, And how it's activated, in part, it's by the cytokines and the antigens displayed by the infected cells. So those two things here, those are these two things right here. So it's binding to the infected cell, and it's the cytokines from the helper T cell. And then they divide and differentiate to make more active cytotoxic T cells, and it also makes them memory cells. So look, you have all kinds of memory cells. You have memory B, memory helper T cells, and memory cytotoxic T cells. So now when we get a clearer picture, when you get a secondary infection, you not only have memory cells that can immediately begin secreting antibodies, you have helper T cells that activate those memory cells and activate more B and T cells. And then you also have memory cytotoxic T cells that can immediately begin to bind to infected cells and trigger apoptosis. And so that's because once it becomes an active cell, that's what it does. And so those are the cytotoxic T cells in your cell-mediated immunity. So those are your two types of immunity. Helper T cells help out with both of them. Different end results, killing off the pathogen, killing off infected or cancer cells or transplanted tissues. All right. There are different types of antibodies. And so we're going to skip back to this B cell. We already talked about it a little bit. There are different types of antibodies. You do not need to know the 5 major classes and what they are, but they're just to know that's antibodies, that there are different classes. And another word for antibodies is immunoglobulins, sorry. And oftentimes they shorthand it. So if you look here, immunoglobulins is oftentimes it's represented by IG, and then this right here will be the type. So there's different types. So IG is the immunoglobulins. So if you see that, that's what it's referring to. And so we talked about the B cells already. So we've already talked about this and talked about the B cells here. So let's add on to this a little bit here. So you have the antigen presenting cell presenting the antigen after it took in the bacteria, activating the helper T cell. The activated helper T cell releases the cytokines, which is going to help activate the B cell. The second thing that helps to activate the B cell is what I want to talk about, what's going on right here. Notice that this is the bacterium. Look, it's the same thing. And so the bacterium can remember, the pathogen can bind to the B cell, because remember the B cell, we wrote on the board that the B cell has a site on the ends of that Y-shaped receptor that binds to the specific antigen epitope on the pathogen. And so that's what's going on here. So let's write what's going on here. So let's write a few things here. The first thing I want to write is that the pathogen binds to specific B cells. The second thing that goes on here that it's not showing you here is that the pathogen takes in some of these antigen molecules, takes in some of these bacterial compounds by endocytosis. So the pathogen takes in antigen molecules by endocytosis. And then what it does is what it's showing you here is that it presents that. And so that's what's going on right here. So right here is going on with number 3 is that the B cell presents only the antigen to which it binds. So So that's like this one right here. So it's binding to it, and so then it presents the one that it's binding to. And so that also helps to activate the B cell. So the B cell is activated by the helper T cell as well as with the binding of the pathogen. Although most of the time, you need the helper T cell to bind to the B cell. So this alone, the binding of the pathogen, will not activate the B cell to trigger clonal selection. You need the activated helper T cells. So let's write that here. Most antigens require active helper T cells. These cells to trigger a humoral response. Okay. So then let's look at this. So antibody-mediated disposal of antigens. So we've talked about the antibodies with the humoral, with the B cells, that they're instrumental in getting rid of the pathogens. So how in the world do they work? And so that's what we're going to look at here. So the binding of antibodies to antigens is also the basis of antigen disposal mechanisms. How do we get the antigen and the pathogen out of, because remember the antigen is on top of on the surface of the pathogen. So microbes are eliminated by phagocytosis and complement mediated lysis. And so I had shown you this picture already, this picture right here. So the complement system. So this is binding of antibodies to antigens and activates antigens by. And so here's your antibodies. So we can make all of our antibodies pink here. So here's your antibodies on your foreign cell here. And so these are antibodies, and that triggers the complement system, which remember are generic proteins that will trigger the cell to lyse by triggering it to kind of poke holes in it, and so water can get in and the cell lyses. So this, remember, is kind of generic part. And then this part right here, the binding of antibodies to the antigens and activates antigens by, these three help for phagocytosis to happen. So your phagocytic cells, like your macrophages, helps them to get rid of your pathogens. So how that happens is, if we look up here, it says viral neutralization blocks binding to host and thereby increases phagocytosis. And so here you have a virus and a bacteria, and here are your antibodies binding it to it. So there's lots of antibodies that can bind to the antigens on the surface of your viruses and the surface of your bacteria and so on. And what this does is these antibodies can bind to the surface of your macrophage and help to phagocytosis to happen, so that you can get rid of those viruses and bacteria. And when all these antibodies are binding to the bacteria and the viruses, that means they can't bind. They're all blocked, so they can't bind to your cells and infect your cells. So that's what it says, blocks binding to host. All right, and so therefore, and then if they can't go into and affect cells, then it gives the macrophages time to engulf them and destroy them. Agglutination of antigen-bearing particles such as microbes. So here's your bacteria, and here are your antibodies. What it does is it kind of clumps them together, all the bacteria together, and so if you have another type of bacteria in here, it clumps them together. That's the agglutination part, and therefore when phagocytosis happens, it takes in multiple living bacteria all at once. In this case here, precipitation of soluble antigens. Let's say the antigens dissolve in water, and most of our fluids in our body are water-based fluids. And so the antibodies bind to your soluble antigens, and thereby notice that there's 2 binding sites on the tips of that Y, and so each antibody can bind to 2 different antigens, and so therefore allows them to precipitate or come out of solution and become a solid in solution, and then the macrophage can engulf them up. So most of the foreign cells are either destroyed ultimately by phagocytosis or this lysis process, and the antibodies aid in those 2 processes happening, hence the essential role of antibodies in fighting an infection. So we had talked a little bit about immunity and vaccinations, and so that's what we're going to talk about here in this next little part, active and passive immunization. So active immunity develops naturally in response to an infection. So this is what we've just talked about. So you get exposed, it activates the helper T cells, which activate B cells and T cells, which then you get memory cells. And so it can also develop following immunization, also called vaccination. In immunization, a non-pathogenic form, so this is why I say make it not harmful, of a microbe or part of a microbe. It doesn't have to be a full microbe. What's important is it has the antigen on it, because that's what your body's recognizing as foreign. It elicits an immune response, and we're just going to leave it like that, immune response. Okay, so it's the same response that we have when we come into contact with a pathogen in, like I said, the real world. All right, so we talked about that. Then passive immunity provides immediate short-term protection. It is conferred naturally when this is a type of antibody. You don't need to know that, but IG stands for antibody. G is one type of antibody. Crosses the placenta from the mother to the fetus, or when, this is another type of antibody, IgA, passes from mother to infant in breast milk. So basically, the mother's antibodies that the mom produces can be passed along to her child when the child, either when the child is in the womb, or that's what I was talking about, it crosses between the placenta when they're still in the womb, or those antibodies can be delivered through breast milk. And so when a baby is an infant, they don't have a fully developed immune system, and so therefore they're not, they don't have a lot of or T cells made yet, and therefore haven't come into contact with a lot of things. They don't have so if they come into contact with a bacterial virus, it can since their immune system isn't fully developed, it can be more harmful. And so breast milk is a way to give a baby passive immunity. Why it's passive is because it's gaining immunity, gaining the antibodies without having a full-fledged developed immune system where they're producing the antibodies, but they're getting the antibodies from somewhere else. It can also be conferred artificially by injecting antibodies into a non-immune person. So in this case here, you're injecting the antibodies. So sometimes if people are going to a foreign country, they may get a shot of antibodies that protects them against a particular disease that is prevalent in that particular country. Those antibodies stay in the body for a certain amount of time, so therefore they're afforded a couple weeks protection against that pathogen that would cause that disease. And so they're not for life immune to that disease, but for a particular time they have antibodies in their fluids in their body, and so therefore have a protection for a while. Okay? And so that is active versus passive immunity. Active is make sure that you get the active cells as well as the memory cells. So active immunity will give you protection for a second infection. Passive immunity, you just get the antibodies, so you get the protection just for a short time period. Okay? And so the next video will be over the next little bit here, so we don't have very much left of chapter 43, and we will be moving on to the next chapter.